Bismillah Rahman Rahim. We start now the MCQs on the chapter 5, which is mitotic cell division. And I hope this can finish in one video. So there is only one video, there's no second video to this one, but I'm just labeling it video 1. Question 1, the photograph shows a stage of mitosis. What would be the correct for the next stage in mitosis? Now, if you know, there is this very typical I have made I for interphase, and then I have a purple mat. So I is for interphase, P for prophase, then metaphase, then anaphase, and then telophase. So very easy way to remember this I, P, mat. What would be the correct for the next stage? You know, this is now the center of the cell. So M for middle of the cell. So M, this is metaphase. So what would be the correct for the next stage in mitosis? So that would be anaphase. So paired chromatose would be absent and the nuclear membrane would be absent. Because you see, the nuclear membrane disintegrated in prophase. And now what they were lined at the equator, not the middle, equator. But I always say M middle, sort of remember it from M middle, but it's actually not middle. Middle means the center of a large sphere, and this is the middle. But this is not the case. The middle here is the equator. This is so they line up at the equator. This is what you need to understand. The diagram shows part of the organization of a DNA molecule and its associated histones. And you know the histones are these complex proteins on which now P is the histones and Q is the DNA wrapped around it, and of course R is just the another protein which is attached to it. So it says which statement were the features labeled P, Q, and R during prophase of mitosis is correct. The linked groups of histones P and R and the associated DNA Q form strands that fold and twist together to form a chromatid. Well, D was the correct answer. The others are all wrong because you see, the coiled DNA molecule forms Q and wraps around the histones of R to form small clusters held in place by histone P. Well, no, that's not. The group of histones P and associated DNA Q move closer together as the chromosome condense around R. No, they condense around the histone proteins P. The histones P and R are made up of proteins around which DNA molecule Q is wrapped so that the DNA molecule can fit inside the nucleus. No, but as it is, it'll fit even when you unwind it. That's what happens. They condense in mitosis, but then they uncondense also. And of course, uncondense is no word. Now, when reading the question, you should underline which row correctly shows the behavior of the nuclear envelope, the centrioles, and the spindle during a stage of mitosis. So they're giving you the different stages: prophase, metaphase, anaphase, telophase. Prophase is when they become prominent. Metaphase, middle. Anaphase means apart, or I say in Urdu, alag. And telophase is there are two nucleuses. T for telo, T for two nucleuses in one cell. So it says which row correctly. So in telophase, the nuclear envelope has reformed as correct. Centrioles one beside each nucleus, and spindle microtubules break down. So D was the correct answer. Then it says which group of cells can all divide by mitosis. Now the first thing to cancel out was bacteria. Bacteria divide by binary fission. They never divide by mitosis. Something you have to know this. This is from the chapter 1. And then cancer cells, lymphocytes, red blood cells. Red blood cells cannot divide by mitosis. There is no nucleus in them. So cancer cells, you're left only with choice D. Then question 5, the photomyritis shows cells in various stages of the cell cycle. Which cells contain twice as many DNA molecules as a cell from the same organism after cytokinesis? So that means the cell, two nucleuses have been formed, and then two cells have been formed with a nucleus in each. Now it says which cell contains twice as many nucleus as a cell from the same organism after cytokinesis. Now prophase, because you see, you've got to remember now the cell cycle. If you remember the cell cycle, so this is prophase, metaphase, anaphase, and telophase. And then we have cytokinesis. And then what do you have? And then you have this whole thing is interphase, in which S phase of interphase, the DNA replication has taken place. So G1, S, and G2. So which now? So this would be prophase. This would be metaphase. This would be telophase. This would be prophase, and this would be late anaphase. This would be more towards telophase. So which cells contain twice as many? All four of them, because these are all still prophase, metaphase, anaphase, and telophase. This is this part, the red part of the cell cycle, but the DNA replication has taken place here in S phase of interphase. So DNA double is double from S phase onwards. This is double. Then a group of chemicals used to treat cancer prevents the formation of the spindle during mitosis. Which phase of mitosis is the first to be affected? So spindle during mitosis. And phase first to be affected. Naturally, prophase, they start condensing and thickening. So then what would happen? That they would not be able to be, they will become shorter and thicker. But then the spindle fiber is not going to be affected. So spindle fiber is not going to form. So they are not going to attach to the spindle fibers. And that happens in prophase. Now question 7. Mitosis is an important process for organism. Which feature of mitosis is important for single-celled organism? So single-celled organism could be any, could be bacteria, but they don't divide by binary fission. So it has to be single-celled organism like protoctist. So it says which features is important for single. So asexual reproduction, yes. Production of genetically identical cells. You see, it won't be growth. Growth is only in multicellular organisms that mitosis is needed. If you and me grow in size, it's by mitosis. But if it's a single-celled organism, well, it's going to divide into just two. So it's not going to become, it's not going to grow in size. We grow in size by adding cells to our body. We grow in length, our bones grow, our muscles grow, nerves grow, blood vessels grow. So growth will not be part of single-celled organism. That is what you had to correlate, and that was wrong. So it couldn't have been two in any of them. So it was 1 and 3 only. Then which statements about complementary base pairings are correct? Cytosine forms three hydrogen bonds with guanine. Purine and pyrimidines are different sizes. Yes, purines is a double bond. Pyrimidine is a single bond. Uh, it it allows transcription to occur. The base pairs are of different lengths. Yes, 80, 80, 80, then GC, GC, GC. So 1, 2, and 3 were correct. 4 was wrong. The base pairs are of different lengths. The base pair A and T and G and C. That's why you get a parallel strand. They're all the same. Purine is adenine and guanine, and they are double ring, and that's a single ring. So the base pairs are of different lengths. Base pairs are of different lengths. Doesn't make any sense. Which statement about a diploid cell are correct? Question number 9. Which statements about a diploid cell are correct? Can divide by mitosis to repair itself. I always tell you that that's the biggest thing that I do all the time. Cell cannot repair itself. Yes, tissues repair. Tissues can repair, like you have a cut, or you have an appendectomy, the doctor cuts your skin and your muscles, and those tissues can repair by mitosis, but not a cell. Many cells joined together make a tissue. So do not know the difference between a cell and a tissue. So it can't be one. So if one was wrong, this is wrong, this is wrong, and this is wrong, you're only left with choice D. Then which metabolic processes will be very active in a cell that has just completed cytokinesis? Just completed cytokinesis would be 1 and 3, because if it's completed cytokinesis, then that means number 1, mitosis has taken place. So PMAT has happened. PMAT is mitosis. This is all mitosis. And then we have cytokinesis, in which the cytoplasm divides. And then we have the whole of it, which is interphase, which is divided into three phases: G1, S phase, and G2 phase. So it says which metabolic will be very active that has just completed cytokinesis. So growth 1, G1 growth 1, in which there is ATP formation and protein synthesis, but DNA replication takes place in S phase, so that is not, that has just completed cytokinesis. DNA replication takes place in S phase of interphase. So DNA replication will not take place, so it can't be 2. So that's why the answer was 1 and 3 only. Now, question 11, which function is correct for the description of the cell structure? Organize microtubules, package hydrolytic enzyme, this would be Golgi apparatus, synthesis of lipids, this will be SCR, and synthesize polypeptides, so it's a membrane-bound spherical structure. Synthesize polypeptides, so ribosomes which are attached to the rough ER. That would be wrong. Synthesize lipids, membranes which surround an enclosed inner cavity. Membranes which enclose an inner cavity. So this is the SCR. So synthesized lipid was correct. This was wrong because membrane bound ribosomes are not membrane bound. So this was wrong. Hydrolytic enzyme non-membrane bound cylindrical structure, that centrioles, no, that was wrong. Organized microtubule centriole membrane bound sacs, no, centrioles are not membrane bound. A student examined, question 12, a student examined the cells in the growing region meristem of an onion root and obtained the data below. Interphase 886, prophase 73, metaphase 16, anaphase 14, telophase 11. What percentage of the cells contain chromosomes that appear as two chromatids? As two chromatids appear. So you add prophase and metaphase, and that's 89, so 8.9%. Which definitions of diploid and haploid are true for typical eukaryotic cells? Diploid, any cell with an even number of homologous chromosomes. Diploid, any cell with two copies of each homologous chromosome. Haploid, any cell with half of the diploid number of homologous chromosomes. Any cell with one copy of each homologous chromosome. When I knew 4 was right, the rest then I would have figured out which ones were wrong as well. So any cell with an even number of homologous chromosomes. What do you mean that? 10, 12. So a cell with even number, all of your cells of the body have even number of homologous chromosomes. Then, so it was 2 and 4. There are two copies of each homologous chromosome. Again, this is more of an English testing exercise. The cell cycle includes mitosis, which are features of nuclear division. Prophase, metaphase, anaphase, and telophase. P, M, A, T are only part of mitosis. Interphase is not part of mitosis. Cytokinesis is not part of mitosis. So forms equal cells of equal size to the parent cell. That would be cytokinesis. So 1 is out. It's wrong. Forms genetically identical cells. Yes. Semi-conservative replication of DNA occurs in interphase, not in mitosis. It's S phase of interphase. Question 15. Which is the correct statement concerning cell and nuclear division? The answer was A. Haploid eukaryotes can reproduce by mitosis, whereas diploid eukaryotes can reproduce by mitosis or meiosis. Correct statement concerning cell and nuclear division. Then question 16. Diploid organisms that reproduce sexually produce haploid gametes. Some plants such as wheat can produce diploid or haploid gametes, and these gametes can fertilize other diploid or haploid gametes. Which statements are correct for plants like these? Diploid gametes may be produced by a fault in the reduction division. Yes. The offspring will always show an increased chromosome number, not necessarily. The offspring could either be 2n, 3n, or 4n. Yes, that's correct. The chromosome number could, in theory, increase in each generation. Yes, that could be correct. Because if a diploid gamete fuses with a haploid gamete, then it's a triploid zygote. You study this in A2 as well. Which statements about cancer are correct? Exposure to UV light from the sun always causes skin cancer. No, not necessarily. Some viruses can cause cancer. Yes, the mutation of genes controlling the cell cycle may cause cancer. Yes, so it is 2 and 3. The diagram represents the life cycles of a sexually reproducing animal. Which row correctly identifies meiosis and mitosis? So meiosis had to be something 2n to n. So that was only 3 was possible. And then of course the number in mitosis must remain the same. So 1, 2, 3, and 4. N has become 2n here. So 2 was the only one which was 2n to 2n. So that's why it was 2 was mitosis. Then coming to question number 19, chromosomes telomeres are essential for DNA replication and are not completely replaced during mitosis. A substance X is known that completely replaces telomere during mitosis. What will be the effect of growing cells with and without substrate cell? So with substance X, cells divide continuously. Without substance, cell division eventually slows and stops. Because as you lose the telomeres, so without substance X, you see, you will have to eventually slows down and stops. The diagram shows the cell nucleus in the prophase of mitosis. Which statement describes the chromosomes found in each daughter nucleus immediately following division of this cell by mitosis? So first of all, let's 1, 2, 3, 4, 5, 6, 7, 8. So there are 16 chromatids. When two cells will form, there will be 8 in each. Which statement describes the chromosomes found in each daughter nucleus immediately following division of this cell by mitosis? 8 chromosomes each, containing one molecule of DNA. So this, when you have this double helix, this both these is one molecule of DNA. These are not two. This one and this one together, they are twist around each other. So this is one molecule of DNA. Then 21. Which feature of mitosis ensures that all the genetic constitution of the cell is maintained? The position of the chromosome on the equator of the spindle, the longitudinal division of the centromere, the DNA of the parent cell replicates before mitosis began with the pulling apart of the chromatin. So one was the only one which was wrong. The position of the centromere on the equator of the cell. So this is a cell. Whether a chromosome is somewhere here or somewhere here doesn't matter because it's going to be pulled. And one will go here and one will go here. Similarly, one will go here and one will go, whether it's here or anywhere on the equator, doesn't really matter. Then question 22, immediately after which stage in mitosis an animal cell does the cytoplasm start to divide? Naturally, as I just told you, I will keep on doing the cell cycle with you all again and again. This is interphase, right? And this is purple, prophase, metaphase, anaphase, and telophase. And then we have cytokinesis. This is cytokinesis. So it is after telophase. After telophase. 23. The diagram shows a cell of an organism formed by reduction division. What is the diploid? This is a cell of an formed by reduction division. So I always say, okay, start marking it. One. No, let's do it with the red color. One. Let me just rub it off once. So you count them and you mark them. One. I'll just do it, and then I want you to see it. 1, 2, 3, 4, 5, 6, 7, 8. Then 9, then 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So this is 20, one of each pair. So what is the diploid number? 14 actually. But you would count it wrongly, and that's where you lose the mark. Each of the following events take place during mitosis. Please see that the year, if you feel the mark scheme is wrong at times, there's one video in which I made one mark scheme wrong, then you just check it with the year and the number of question. Each of the following events take place during mitosis. Centromeres divide, chromatids move to opposite poles, chromosomes line up along the equator, metaphase, chromosomes uncoil, two chromatids are joined by a centromere. So in which order do the events take place? First of all is 5. The two chromosomes are joined with this centromere. That's in the prophase. Then number 3, chromosomes line up along the equator, metaphase. Equator, metaphase. Then anaphase would be 1, centromeres divide. The centromere divides. This separates. The chromatids now separate. They're no longer called sister chromatids. Now they're called chromosomes. And then, of course, was 2, which was chromatids move to the opposite poles. And number 4, chromosomes uncoil. They sort of then again become thin thread-like back to interphase. In interphase, you can't see them prominently. They're just a dark staining nucleus. And because you cannot see them. But in prophase, if you see the nucleus, you can actually see the chromosomes because they've shortened and condensed. This is prophase, and that's why I say prophase. P for prophase, P for prominent. They have now become prominent. They become shorter and thicker, chote and mote. Diagram shows the cell cycle. When radioactive nucleotides are supplied by dividing cells, at which point will they be incorporated into the chromosomes? So you have to see that B and C are the early mitosis, then D would be cytokinesis, and A would be part of the interphase. That's where the DNA replication is going to take place. 2026. Which statements about a diploid cell are correct? Can divide by mitosis to repair itself. Cells cannot repair. We've done this question earlier on. 27. Meiosis and meiosis are two types of cell division. A cell has 20 chromosomes before it divides. How many chromosomes will it have after dividing by meiosis? So after meiosis, it will be 10, and after mitosis, it will remain 20. The graph shows the length of the spindle fiber during mitosis. Which region of the graph shows when the centromeres detach from the spindle fiber? Now, this is technically a very, very good question. Length of the spindle fiber. So now you got to imagine this cell. You got to imagine this chromosomes here, and this is here. And then you got to imagine here is the centromere, right? Shows the length of the spindle fiber. The spindle fiber is attached here. Now let's look. I'm going to give another color to the spindle fiber. This is the spindle fiber. So as this will become shorter because you see as they move to the poles, so they're going to move to the poles. And then look at the next stage. They become even shorter. So here they're attached to the centriole. So it said which region of the graph shows when the centromeres detach from the spindle fiber. So you see length of the spindle fiber. Watch it here. Look at this length, 1. Look at this length, 2. Look at this length, 3. These are the different lengths of the spindle fiber. So which region of the graph shows when the centromeres detach from the spindle fibers? You see, you can see them that they have now left it. First of all, the length of the spindle fiber is increasing. So this was the time. And then it is decreasing. And so it would be the D part of it when the centromeres detach from the spindle. And that's why they are now then, you see, then they have moved to the poles and then the new nuclear membranes will reform. The graph represents the changes in the quantity of DNA present in one nucleus at different stages in the life cycle of a mammal. So you see, originally there were 2. Now it's gone up to 4. DNA replication has taken place. And then you go back to 2. And then, so what has happened here is telophase, because you see it was one cell with 2 nucleuses. And now there are 2 different cells with 2 nucleuses. So what happens is first from 2 to 4, that means sperm and ovum fuse to form 4 arbitrary units of DNA. And then fertilization has taken place and now there are 4. And then the 4 becomes 2 again. So that means our cell is dividing quantity of DNA present one at different stages of the life cycle of a mammal. So from 4 to 2 means that cells are undergoing mitosis. So this is at telophase, because in telophase now it says DNA present in one nucleus, not in one cell. Because please remember, in telophase This is one cell, has 2n here and 2n here. So you have 4n in telophase. And then, of course, cytokinesis take place and the two cells form. Then question 30, at which stage of mitosis do these events occur? Centromeres separate, spiralization and condension of DNAs, anaphase and prophase. Diagram shows the chromosomes of a typical plant cell at the metaphase stage of mitosis. So you see what they've done is they've taken the cell, and this is where they've aligned at the equator. And what have they done is they have cut it from here and they're showing it to you from the top. You're looking at a cut orange from the center. Which row describes this cell during metaphase? Diploid number for the plant, 8, cell wall, metaphase, centrioles. There are no centrioles in plant cells. And spindle, of course, is present. So that's why the answer was C. You must number them and see that they were actually 8. So diploid number for the plant would be 8 because at the moment they are 8 to the 16. There are 16 chromatids, but there are 8 chromosomes. The diagram shows chromosomes at metaphase of mitosis. What are the diploid and haploid numbers? Again, just count them 1, 2, 3, 4, 5, 6, 7, 8. So diploid would be 8 because they're actually there are 16 chromatids, and haploid would be 4. But you have to be understanding this. What occurs in anaphase of mitosis? Chromatids separate and move to the opposite folds. Anaphase, alag, apart. 37. In a DNA molecule, the base sequence AGT codes for the amino acid serine. What is the base sequence of the anticodon of the tRNA? Well, that was AGU. So this is a very simple way that you do it. You say DNA, mRNA, and tRNA. Of course, it's not part of this chapter. This is part of the next chapter. So the DNA was AGT. Done that, you write the DNA and then you write the mRNA, and you know the mRNA and the tRNA will have U, so there's no thymine. There's no thymine. There's uracil instead of thymine. Then last question, what is the minimum number of base substitutions required to change the nucleotide sequence of the HbA normal allele to HbS sickle cell allele? There's only one that was substitution changes, but I think this has been removed for the new syllabus. That finishes this chapter, and I hope these MCQs are helpful, and I'm probably going to get some more new questions and do another video in a few days. So hopefully, yes. And thank you very much for subscribing, and thank you for liking the videos, and thank you for all the very nice comments that you give me, and it makes me very happy, and I feel I can go on and do more and more videos for you so that you all can benefit. And thank you very much once again.